Okay, hi everyone. Welcome to the seventh seminar in the Computer Engineering Seminar Series. I'm Tushara Bandara, who is going to coordinate today's session. Here, our speaker is Dr. Hasindu Gamaarachchi, one of the graduates from the Computer Engineering Department in 2015. Yeah, I think most of you know him very well. Okay, anyway, to give a brief introduction about him, actually, as I already mentioned, he received his bachelor's degree with first class honors for computer engineering from the University of Peradeniya. And then he entered the University of New South Wales, Sydney, and received his PhD in computer science and engineering in 2020. And currently, he's working as a genomics computing systems engineer at Kinghorn Center for Clinical Genomics in Australia. Yeah, so that's a bit about him, right? I'm not going to talk any further because it consumes your valuable time. Instead, I would like to hand this session over to Dr. Hasindu Gamaarachchi to continue from here to talk about his findings and to share his experience with us. Over to you, Dr. Hasindu.
Okay, thank you. Hope you guys can hear me and see my screen. Is it shared?
Yes, we can see your screen. Yeah.
Yeah. Okay. So like Dr. Damayanti invited me to talk about this, like Genopo, one of the like softwares we did to enable sequence analysis on portable Android systems. And I thought of like giving the background to it while focusing on how as computer engineers we could like help like various multidisciplinary projects to do things with that you couldn't probably do without that knowledge. So I would like to keep the conversation a bit interactive if possible, because that way it's more like interesting if you actively engage. So please like feel free to like interrupt in the middle and ask questions. And also if you couldn't understand something, please always let me know so that I can repeat. To start with, let me give you an introduction to what these genomes and sequencing, those terms are, because most of you are like not familiar with biological side for sure. So let me do that first. Okay. We all like know that like living beings are made of these things called cells. And let me get the laser pointer. Okay. Cells, and inside the cell, in the nucleus, there is this thing called like the genome. Where in most mammals, like humans, this genome is made up of molecules called DNA, whereas in viruses, it is RNA. So these DNA molecules, few of them makes up the human genome, the humans. The interesting thing is, this is like a blueprint of life. All the information necessary to make a particular individual from the scratch, and also like how the living being would function throughout the life is coded into this like DNA. We can think of it as like a computer program, where in a computer program we store all the instructions and the data required for a program to start and run. And we do that by storing those instructions and data in binary using ones and zeros. Whereas in DNA, this data is stored as a series of 4 letters, namely A, C, G, T. So these ACGT are like representations for these 4 molecular units. So this DNA is like a long molecule made up of these ACGT bases. When you like represent it in letters, it's a long string made up of A's and C's and G's and T. So the human genome is totally like 3.2 billion bases. So to write it as a sentence full of A's and C's and T's and G's, they say that it will span from Singapore to Australia. The font size, I don't know what they measured that. So, so this is like very important in many fields. Any fields like where we are going to like involve any organisms, knowing about the genome is important. Obviously for humans, for healthcare, like providing precision medicine, and on the other hand, like to enable, like to know what will happen to you in the future so that you can take precautions, whether you are like prone to a certain disease, so that you can take precautions before it happens, because we all know that prevention is better than cure. And also these days, this is becoming extremely like useful to like because of the ongoing pandemic, because DNA sequencing gives us the opportunity to like study the virus and so on. And also in agriculture for like various crop improvements and stuff, forensics. So any field where like organisms are involved, the genome study is important. So now let us see how this DNA molecule is read into a computer. Because I told you that this is a long molecule which is stored inside the nucleus of the cell. And each cell in the body has a copy of the DNA. Now this figure, which I just copied from like National Human Genome Project, it shows like from a living tissue, in this case the liver, you get some like tissue or blood, and then you apply some chemicals to break the DNA, break the like the nucleus and get the DNA outside, and then they like apply various chemicals to like make this like untangled, because it is stored in a very like, you know, like maybe like micrometer scale, so you need to like make it untangled. So they chemically do it, and during that process these DNA strands break into little pieces. So and then like they do a step called PCR, where they like multiply all these kind of like little pieces. Basically they make thousands of copies from each, and then they put into the sequencer, where they will use some like light-based or current-based censoring technique to like read those fragments. And those fragments, which are composed of A's and C's and G's and T's, would come onto the computer. So when those come, you have to do a number of computational analysis steps to interpret some meaningful information. The reason is, as I told, like this process will break the DNA strand into pieces, so from random locations. So what we get through the sequencer is a set of fragments with we don't know what the order is, from a random order. So you can think of an analogy like this. Let's say you take a paper, like some like A4 paper with some sentence written. Okay, you like tear it apart into tiny pieces. You randomly shuffle them, and then let's say you make some copies out of each piece, how I do that, I don't know, but let's say you make a set of like copies and then again shuffle it, and then you randomly pick one and then write whatever the content and take another one, write the content and so on. You do it for each and every piece until you get enough data. Now, from all these pieces that you gathered, you need to reconstruct the original, like the writing. So this is what actually the analysis. So that's a very complex process, because you could see that it is not like a simple thing when millions of pieces in random order are like here and there. So I'm not going much into like details, but we'll like go to like more computer engineering perspectives from now onwards. So if you talk about like the history of DNA sequencing, like in year 2000, the DNA sequence looked like this. Whereas like a couple of years ago, like this, not maybe like 5 years ago, these DNA sequences have shrunk into a device which is like handheld. So the DNA sequences have moved from this much large size devices to small size devices. They are ultra portable. But the thing is, still the scientists use these supercomputers to process the data. What I mean by supercomputers is like these rack servers made like a lot of rack servers. Like you guys might be using the Icon server on the department. So basically, a supercomputer is composed of like hundreds of such blade servers. So that's how they usually process the data coming from even this little sequence, because the data size is like hundreds of gigabytes in size, and the analysis algorithms that do that putting together thing is very like time consuming. So there's a problem, because in many cases where this ultra portable sequencer could be used in the field, still the data had to be transferred. For example, several years ago, there was like a Ebola outbreak in West Africa. So at that time, the scientists had to like send the data from the like epicenter or whatever in the like pandemic to UK by internet to like process the data on a supercomputer. So this limits the actual use of a portable sequence, because the data transmission, as you know, terabytes of data is not something very easy to do via like remote regions like West Africa. And another example is where the sequences were taken into space stations to study astronaut health and stuff, but still the data had to be brought onto the Earth to process. Likewise, there are many examples. So several years ago, like when I started PhD, was to see whether we could make ultra portable genome analysis devices to make use of these ultra portable sequencing units in like a real, like to make real use of it. So when I started like this project, yeah, it was a bit of a controversial like topic as well, because many like scientists like think that these algorithms are extremely like, you know, time consuming, computationally intensive, data size is big, so that's why they are like done on like a server, and it will not be really feasible to do these kind of like analysis on portable devices. But at the same time, we as computer engineers know that today the like our laptops, mobile phones have some decent amount of computing power. But first of all, let's see what kind of challenges lie in making these algorithms that run on supercomputers to run on like a order. Realize by now the tools are very complex. It is not just like dozens of algorithms which are stitched like in various order so that you get something useful. And also there are many heuristically determined parameters for the computations which has some meaning for the biologists. So we can't like change much around these like parameters or algorithms because like a very subtle change can affect the accuracy. And accuracy is a very important thing in DNA analysis because, as you may know, like if you give a slightly different output to the most accurately possible, that might like have an impact on the decisions made by like a clinician or like whoever. So this is the challenge like that I found the most. But also as like a computer engineer who know how like the computers work, I could like see the algorithms, like how they are implemented, and realize that the biologists, like the computational like biologists who wrote this software, they write software to solve a certain like a biological problem. So they don't really worry about how the computer is built or whether like how various operations happen inside. So they have access to these huge computers in their research centers with terabytes of RAM and like large number of processing cores, and they usually like have almost infinite computing power and RAM. So they are not really worried about writing optimized code. They just write assuming that they have unlimited computing. So as like someone who know like how a computer really works, it's an opportunity to optimize. Of course, there have been some previous like works that have focused on accelerating these like analysis software, but most of them had like focused on little pieces. I told that these like software pipelines are made up of dozens of algorithms, but most of the previous work take just one component, accelerate that, and show that, oh, this is this much time faster. But the thing is, it won't be really used by anyone because it's independent. For a computational biologist, for really make use of your optimizations, you need to integrate it to an actual workflow and provide them so that they can really use it. And when you integrate also, number of challenges that affect the performance might come. So to start with, we built like two sets of like devices. So this device was, the one in the left, was made out of 16 single board computers. Like you may know like things like Raspberry Pi. This is like this was a better version of a Raspberry Pi. It had like 4 GB of RAM and like 4 ARM cores. And also like after a while, we made one out of these Jetson nanoboards from NVIDIA. So each one has like ARM cores, 4 GB of RAM, and also a GPU. So the idea was to optimize those, one of the software pipelines for DNA analysis to work on this and this. So now let's talk in terms of what we have learned in computer engineering courses so that we can like see how we can apply these. So we know that the modern computer system is made up of a memory hierarchy. This is the picture I just got from somewhere, so it's a bit old. So you can see like today you don't have magnetic tape. So now you can replace this magnetic disk with the SSD and the tape with a disk. So registers are the fastest, and likewise, when you come to traditional hard drives, it becomes the slowest. So we all know that why we have this is most of the code, we assume temporal and spatial locality. When you access data, like we assume that most of the like accesses would be either ones which were accessed just before or something close in the memory. So this is something really important that we need to like consider when we are optimizing such like code. For example, let's take a situation where you can go through a series of data from left to right. There are like different ways to implement. One method might be to put it into a linear array and then sequentially access. Another way might be to put it into a hash table and then based on the index you can access. From a biologist perspective, they won't see a difference. They might think the hash table is probably easier to use. But we as people who know have learned the underneath implementations, we know that accessing an array sequentially can be dozens of times faster than accessing random places in the memory, because all these caches and like buffers come into use when you sequentially access. That's one example. Also, you may have learned in various courses about multi-threading. We all know that like if you just write a multi-threaded code without really knowing how bottlenecks can come, there won't be any really use. Just spawning some threads and then processing doesn't really do any. We need to make sure like there are no bottlenecks, especially when you are like wrapping something with a semaphore mutex. We have to make sure what's happening inside that like critical section is really less. So unnecessary part you shouldn't like put into those sections. But if you look at some code written by a biologist, like they just wrap huge pieces here and there. And also another example is accessing the disks. So especially these magnetic disks, we know that we should minimize access as much as possible. If you can keep it in the memory for a future use, that would be really beneficial, because going and accessing something from the disk is pretty expensive. And also, we know that the way the magnetic disks work, once you start reading, if you sequentially read, you can reach perhaps like several, like 100 megabytes per second. But if you do random accesses, it will drop to maybe a couple of megabytes per second. So writing your software, restructuring it in a way that these features are really used is quite important. So I'll show you like a simplified workflow, how you can like take a piece of software written by someone and then nicely optimize it. First, you can analyze the workflow by using various profiling tools. So just putting timestamps. And then we can identify different types of workloads. Some of them might be processor intensive, some might be IO intensive, and so on. And then you can systematically restructure these. And then you can like optimize the bottlenecks by taking into consideration all the important, like, you know, computer architectural features we know of. So if you do that, you can make sure we use those resources very effectively. Even if you have a very powerful compute system, if you don't really write efficient code, it won't be effective. But if you take these into consideration, we can make sure the utilization is quite. But there's another part to this. We should also take into consideration about these characteristics of biological data and algorithms, the heuristics and parameters. You can't mess up around those, because if you do changes to those, then it will impact accuracy and the optimizations will not be useful. So if you keep those two factors, then you can make sure you get some efficient piece of software that will run on an embedded system. So this kind of analysis and optimization, we I first applied to a popular analysis workflow, something called methylation calling. I wouldn't go into much details. So it is made up of 5 pieces of software. I'll focus on the two the major bottlenecks. The first one was an alignment algorithm, which requires a massive amount of memory to process. But by using some divide and conquer method, we were able to reduce the peak RAM capacity. So the original software required 12 GB of RAM, which is not available in most embedded computing systems. But by carefully analyzing what's happening inside and factors affecting the accuracy, we could bring down the RAM quite a lot. And then this methylation calling software was very time consuming. But when you really analyze the software, you can see even though it is multi-threaded, not all the cores are effectively used because of various bottlenecks. So we restructured this whole thing, rewrote the framework, and also then did some GPU optimizations, and we were able to bring this bottleneck to a very good level. There was a huge performance improvement. So then after all these kind of optimizations, it was possible to achieve processing a data set on this undistributed cluster within 5 hours. Whereas with the original software, not the optimized one, if you run on a high performance computer like the Icon, it also took a similar. So the reason is, even though there are 64 cores, because the way the biologists have written the software, it's not really used. Multi-threaded implementations, most of them either are stuck at disk accesses or memory allocation bottlenecks. So not really effectively used. So that's why we could achieve similar performance on a distributed cluster like this. And also with GPU optimizations, we could make it this little Jetson device capable of processing the data from a sequence in real time. When you plot a sample and run, the sequence like this would take 50 hours or 48 hours to give the output little by little. So you can plug in a device like this directly, and then if you implement if you come up with the real-time analysis method, you can keep processing, and then can you are able to keep up with it. So that was what I was doing during the type of like the period of my PhD, just a very like high level recap without going into very like algorithmic or technical details, because those will take a lot of time to describe. So simple. And then when we like showed this to like, okay, look, like these things can be done on this. One question that came into our like mind was, can we do this on a smartphone? Because from a computer engineering perspective, we know that a board like this, a Raspberry Pi, has the kind of the same like chip, RAM, hardware, like to a smartphone. So in theory, if you show that something works on a system like this, that means it can happen on a smartphone. But from like the scientist's perspective, they don't really like see the difference. So we needed to we wanted to just like implement some like app to show that you can do these kind of analysis, not just on a little device like this, but also on a smartphone. So like in 2000, I think 19, like with Dr. Roshan, like we put a final year project for Peradeniya computer engineering, and you may know these three guys, Hiruna, Jana, and Sanoj. I think Hiruna is even here in the talk listening. So we put a project for like doing this, taking the things written for like this to get run on Android like that. So many challenges. I remember like Hiruna said, like perhaps like we could get this within like few months. But on the way, like many challenges came into the project. One major like challenge was because these softwares keeps on updating. When some like bug or something discovered, when the new data arrives, sometimes you have to do some modifications. So any such modification needs to be quickly integrated into the software. So you can't just compile and you can't just do it once and just say it's done. You need to come up with a way so that those changes can be quickly integrated. And also the way the Android operating system works is a bit different to like a normal Linux system in terms of security and containment. So to circumvent those, you have to follow what's given in the Android toolkit or whatever. So, but Hiruna and others came up with a very good architecture to solve these problems. So as you can see, it's a mixed number of systems engineering steps where you make a shared library out of the tool and then use this thing called the native interface to link to the GUI, which is here. So the GUI looked like this, and the performance was quite good, which we did using a little human genome dataset on very some cheaper phones. And then we were thinking, we were just drafting a manuscript at that time, and then suddenly this coronavirus also came, and then we thought, okay, given that we made it in a way that it's very easy to be modified, let's implement the coronavirus analysis workflow as well onto it. So because it was implemented in a way that it can be quickly adapted to other tools, it was a very convenient job, and we could show that on several smartphones, you can do a coronavirus analysis within processing can be done with like 20 or 60 minutes, depending on the phone's capacity. So this one then, like, because of the use case, it had a good impact, and we were able to finally publish in Communications Biology, and there were a bit of like attention from the media as well, because it sounds like a cool idea where you can use a phone for something that previously required a supercomputer. So that's like a very high level overview of the work that we have been doing and various opportunities that is there for like computer engineers in various other scientific fields. So now, like, I would like to give you the opportunity to ask questions. Don't know whether like I was too fast or like, but if there is any place that you want clarification or doubts or any other thing, yeah, please feel free to ask. Don't be shy.
I have while while others are getting ready for questions.
Sorry, sir.
While others are getting ready to ask question, let me ask one.
Okay, sure.
So, what's the next step? What sort of things you are explaining about this project? What else can be done?
In terms of Genopo,
Yeah.
I think Hiruna is also there. So yeah, so I think like Hiruna, then others, they were like trying on like different like multi like device related things, which we really like, I think, complete to a very end. And also there are other real time like processing opportunities in this area. And also things like base calling, which is currently not implemented into the mobile phone, can be implemented. And likewise, there are like different possible paths that can be, I mean, made into if necessary.
Maybe you should explain from a user's perspective, not from an engineer's perspective.
Sorry?
Maybe you should explain from a user's perspective, not an engineer's perspective.
Yeah. So one idea might be to make use of GPUs on the mobile phones. They are a bit different to like the normal NVIDIA cards, but using OpenCL or something, we could probably try to use them for like real-time base code, because Genopo from some time saying that in the future they are going to make a device that can plug into the phone directly. So in a case like that, having a base caller which converts that current like sensor input into these ACGTs would be pretty beneficial.
Hi, Hasindu.
Hello,
Thank you for that nice visual walk through your project, what you are doing, starting from the basics and going to what you have been doing and what you are doing. It's very impressive work. Pleased to see Dr. Chandana Gamage from University of Morato and Dr. Ruan Veerasinghe from University of Colombo School of Computing joining us for the seminars. So yeah, that's very nice to have you with us today. And I have two questions to ask. One is that, is it how large could the genome size be for that, so that it can suit this workflow? Is there a limitation on that, the species that you can analyze?
Yeah, so so the idea was to like use this real-time capability of the sequencer. Like it will keep producing data, but if you could like process it in real time, then you don't really need to store that like big amount of data. You can directly like go to the final result at the end of the sequencing run. So we can like span the processing over the sequencing run, which usually takes like 48 hours. But obviously one challenge one might go into is the battery life of the phone. But for a very small like genome, like the coronavirus or a bacteria, like can be like done in like 1 hour because their relative data sizes and the processing like requirement is quite low.
Right. And so in addition to that, I was wondering, so I think Oxford Nanopore is a very recent sequencing technology that is coming up.
Yeah.
So for the laboratories across globe, maybe how feasible it would be for them to use it? Because I know someone who is now looking at, for example, my co-supervisor, he's working with sequences of viruses, and he goes to places like Antarctica, and he does sequencing. But one thing he says is that as a biologist, yet maybe he's not yet having these facilities to go for this kind of a technology. So what are your thoughts?
Yeah, so there's like a bit of a gap in that sense, because most of the biologists, if they are like used to the previous like second generations and stuff, they are thinking in terms of that. They rely mostly on the ACGT, like the fast bases that you get. But in nanopore, the base calling accuracy is not that high like in previous Illumina technologies. So you need to like make use of the raw signal data as well during the processing. There are like software and algorithms that does it, but many like try to rely on like old software and they end up like not getting like a very accurate result. But if you like look at the state of the art like software, they are shown to like able to reach like very good accuracy in especially in viruses and all those find mutations, and much like easy as well because of the lack of there's no need to have big capital infrastructure for these kind of things.
Hasindu, you can also maybe mention that we are setting up a few of them here as well locally.
Sorry, sir.
We are setting up a few locally as well. It's not big infrastructure. You can mention about them as well.
Oh yeah, so I think we were, I don't know, like the current progress, but like with medical faculty, like Peradeniya, we were like trying to like get nanopore like sequencing set up like they are to like do coronavirus like sequencing, and also I think Hiruna and all were like working like the agri department were also like thinking about that. I don't know the progress of that, but like I think soon it will like come to a reality. I think the medical faculty one is quite close to that. They are like kind of like in the process of like paying money to Oxford Nanopore. So I think they will soon like be able to do some sequencing.
Thank you again for a nice talk. I was just wondering how difficult the process is. Say you have a certain number of common workflows you have already optimized. If there is some other workflow that is
there are like so many workflows. This is one like workflow for something called methylation calling and doing variant calling on viruses. Those two.
So how difficult is it to get one of those and then optimize the way you have done? Is it a completely manual process? I mean, in the sense analysis and everything is needed.
I mean, like you need to like manually, like, I mean, go through, you can make use of like profiling tools to like see whether you can pinpoint some bottlenecks. But most of the time they are not able to give a good like result when there are like IO related bottlenecks and multi-threading inefficiencies. So most of the time you have to go to like the lowest level and yourself put some timestamps in the code and then do a profiling manually and then find out. And another thing is these codes are like pretty like long and they are made of like number of custom libraries stitched here and there. So you need to go through a complex code base to like get something out of it. So I would say like it's like getting a complete workflow to run on a thing would take a few, like maybe at least a year.
Thank you. So, but I'm really interested to know whether there is any plan to like, make it a kind of a community project. So that people can do that. Obviously that requires a lot of work. And there may be people who are motivated to do that kind of work.
Yeah, I think that's like a good idea as well. Yeah, I think one like obstacle is like in order to like do those modifications, like you need to like first learn some like biological principles and these like things that are important for them, because if you don't like keep those like features as they are, then biologists like won't like really be interested. They are very like serious about the like even like a 0.1 accuracy. So I think like if that challenge could be overcome, like the knowledge, like the domain knowledge necessary from the biological part, then some community project might be like really possible.
Good point. Good point.
Do you have anything to add, Hiruna, if you are still there?
Hi, Hasindu. Hi, everyone. No, actually, you talked a lot. Yeah, that's all about it. So I actually had to learn how nanopore sequencing works and a lot of terms the biologists use, and it was really beneficial for me to work as a research assistant before starting my PhD for about 8 months, where I learned a lot of biological stuff, what they are doing, and also like the wet lab processes, how DNA sequencing happens in the real world, like what chemical things you have to do. I actually saw DNA, how DNA is extracted. So that kind of experience is really kind of useful to start this work. Yeah.
Yeah, I think like there are like, it's a very interesting area because it's really useful, and at the same time, like there are like challenges. Like we will need to like learn another domain so that you can really do something useful, something that really new to you. So you should be able to like have some self motivation to go and like read and understand all the terms and be able to like face all the challenges like that comes. And also the way like biologists think are a bit different to how the computer scientists think. So things that are important to us are not really important to them, and vice versa. So you need to like handle like both sides in a way that it leads to something useful.
I was wondering whether, so you have done some workflows. I was wondering whether, since one of the differences, well, a lot of the tools that are there, bioinformatics work on so-called short reads, right?
Yeah.
And nanopore has long reads.
Yeah.
So I was wondering whether there is some work you can, not you personally, but I mean, we can envisage where you kind of have that layer, you cover that layer so that you can use existing, not on a phone, I mean, on computers, the tools that they have now can be used with nanopore.
That's not really like that easy, because the data characteristics are like completely different. So because like those algorithms are made to work with little like pieces, like reads are like 100 bases, and each base has an accuracy of 99 points. Whereas nanopore has like very like variable reads, like starting from 1,000 bases to maybe 1 million. And each base has a accuracy of only like 95%. So because of these differences, the algorithms are like kind of very different. So those tools cannot be that like easily like changed to work with this. So still like they are like two separate kind of sets, I would say. Even mapping like the state of the art short read aligner is BWA. It seemed to not work well with Oxford nanopore reads, whereas things like Minimap work well, and not vice versa.
Okay, if we don't have any further questions, it's time to wind up, right? Okay, before we wind up, I would like to thank Dr. Hasindu Gamaarachchi on behalf of the Department of Computing Engineering and those who have participated in this seminar. And I also appreciate your time dedicated for this session to share your experience and valuable information regarding your findings, actually based on Genome and Genomics computing. And also thank you all the others for participating in this session. Your active participation is highly appreciated, right? Okay, that's it. So Dr. Hasindu, shall we wind up?
Yeah, sure.
Okay, thanks a lot.
Thank you.
Thank you, everyone.